Throughout the course of history, life evolved from single-cell organisms to the complex life we see today. In this video, we will explore the hurdles that had to be taken in order for life to evolve the way it did. Energy in organisms is generated by rearranging chemical bonds, usually those in sugars and oxygen. When this rearranging happens, the energy in the form of ATP molecules can't be transported anymore. It has to be used instantly. So in order for bigger organisms to exist, a way had to be developed for sugars and oxygen to be transported to where energy was needed. To that end, a system of canals inside organisms evolved, the blood vessels. The blood vessels are filled with water, which in turn carries the commodities of energy. Energy could now be transported to where it is needed, and energy access wasn't limited to the outer cells anymore, so bigger life could be supported. There is, however, a snag. Although this was a great solution for sugars, as they dissolve well in water, oxygen is an entirely different story, because water can only carry minute amounts of oxygen. The total amount of energy transported was fairly low. In order to satisfy the energy need of complex organisms, a way was needed to transport more oxygen than could be carried by blood alone. To that end, a new type of cell evolved, the red blood cell. The main function of the red blood cell is transporting oxygen-carrying proteins. Evolutionary, the first of these proteins was the myoglobin. Later, the more complex hemoglobin evolved. That's the one we find in our bodies today. These proteins are able to transport oxygen much more efficiently than water, or blood in this case. Now, oxygen can just as easily be transported as sugars by the blood. The way the oxygen and sugars are absorbed by the body is also quite different. Sugars are consumed in the form of food through the gastronomical system. There, the food is decomposed and the nutrients absorbed through the gut's membrane. Oxygen, though, doesn't exist in solid form, so it requires a different approach to be consumed. The act of taking up oxygen into the bloodstream is called respiration. Respiration occurs in all shapes and forms, but follows a few basic principles. We humans only respirate with our lungs through breathing. When we breathe, oxygen-rich air is sucked into our lungs, where the air disperses through branches to the alveoli. The alveoli is surrounded by capillaries. In the alveoli, the gas exchange happens and oxygen is transported to the hemoglobin on the red blood cells. The exchange of oxygen and carbon dioxide occurs through a process known as diffusion. To quote Wikipedia, diffusion describes the spread of particles through random motion from regions of higher concentrations to regions of lower concentrations. Unquote. In other words, if there is a small patch of some gas, like oxygen or carbon dioxide, in a corner of a room, provided there is sufficient heat, the gas will disperse through the rest of the room. In the body, this works in a similar way. The oxygen-rich air on one side and the oxygen-depleted blood on the other mix, or rather, their oxygen and carbon dioxide mix, as those molecules are the only ones small enough to pass through the alveoli membrane. Let's take a closer look at the inner workings of the hemoglobin. The hemoglobin consists of 4 protein chains. In adult humans, these chains are 2 alpha and 2 beta chains. Infants have fetal hemoglobin and instead of beta contain gamma chains. The most important job of a chain is to hold a heme group. The heme group is responsible for binding the oxygen. Each heme group can bind exactly 1 oxygen molecule, which makes the oxygen carrying capacity of a single hemoglobin protein 4 oxygen molecules, or 8 oxygen atoms. The overall oxygen transporting capacity of our blood increases by a 70-fold thanks to the hemoglobin. A massive 1.36 milliliters of oxygen can be transported with only 1 gram of hemoglobin, which is a lot better than the 3 milliliters oxygen 1 liter blood can carry through dissolving. Now the body can absorb a massive 1000 milliliter oxygen per minute. This allows our body to absorb 1000 milliliter oxygen per minute. The heme group is the special molecule which is accountable for the absorbing capabilities of the hemoglobin. Thanks to an iron atom in the middle, oxygen is reversibly bonded through oxidation. The iron atom at the center of the heme group is held in place by the rest of the heme molecule, which in turn connects to its protein chain. The first oxygen to arrive at the hemoglobin protein doesn't bind easily. It has to find a way past the protein chains to arrive at the heme group. But when it does, the hemoglobin responds in a very interesting way. The newly bonded oxygen changes the protein's spatial configuration. The protein changes shape. Due to the shape-shifting, the rest of the hemoglobin's chains open up, making it easier for oxygen to find its way to a heme group. Oxygen can bind much easier now, allowing the hemoglobin to fill up quickly. We will see later why this is such an interesting property and why it's beneficial to the body. In order to understand, you first have to know something about oxygen binding. When we talk about binding, we are talking about binding in a macro sense, meaning that if you look at a lot of molecules, you could say they are all kind of bound. On a micro scale, however, these molecules don't actually bind. In fact, they form an erratic relationship, constantly binding and unbinding with each other. Usually, this erratic nature of molecular binding doesn't make that much of a difference. No one is ever going to look at a single molecule. In hemoglobin, however, it does. And for the following reason. Because of the erratic binding nature, oxygen molecules constantly bind and unbind with the hemoglobin. When there is a lot of oxygen around, it does not matter, as there is enough oxygen around to fill the empty spot. If, however, the hemoglobin enters an area with sparse oxygen, the rebinding of the oxygen after the unbinding doesn't always happen quickly enough. If at one point all the oxygen happen to be unbound, the hemoglobin closes again, preventing the oxygen from rebinding. This means that as the oxygen around lowers, the hemoglobin start to unbind their own oxygen. Why this is great for the body is obvious. Where oxygen is consumed, the hemoglobin automatically replenishes the oxygen levels. The amount of oxygen around is called oxygen pressure and expressed as PO2. We are not talking about pressure you know from physics, rather about the amount of oxygen that is present. If you now plot the oxygen pressure and the oxygen bonded by the hemoglobin, you can see how much oxygen the body binds at a given oxygen pressure. The resulting curve is called the oxygen-hemoglobin dissociation curve. The resulting curve is called the oxygen-hemoglobin dissociation curve. If we take a look at this curve, we see that with the pressure of just 50 mm of mercury, the hemoglobin saturation already reaches a very high level, 80% to be exact. Thanks to this early high, adding more pressure won't change the saturation all that much. Even when the pressure is at its normal 100 mm per mercury, the saturation has only risen to 95%. Even when the pressure is at its normal 100 mm of mercury, the saturation has only risen to 95%. It becomes clear that the oxygen pressure of your lungs isn't all that relevant, meaning that with oxygen-depleted air or a damaged lung, you can still absorb enough energy. On the other hand, one can see a sudden drop of the oxygen-hemoglobin dissociation curve at a pressure between 40 and 20. At a pressure of 20 mm of mercury, the saturation is as low as 30%. This is equally important, as this is the oxygen pressure found inside the blood vessels around oxygen-depleted muscles. The low pressure causes the hemoglobin to release its own oxygen molecules so that the muscles can absorb it. There are other things we can explain thanks to this oxygen-hemoglobin dissociation curve. You see, there are various other ways to shift the curve. Shifting to the right means more pressure is needed before oxygen is bonded, while shifting to the left means less pressure is needed. There are various factors that contribute to the shifting of the curve. First of all, there's the pH value. A lower pH, or more acidic, shifts the curve rightwards. A high pH the other way, to the left. This is useful because carbon dioxide, the waste material of oxygen burning, is acidic. So whenever oxygen and sugar are burned, carbon dioxide is created and the pH level goes down, so hemoglobin releases its oxygens. Another waste product of oxygen burning is 2,3-DPG. This molecule affects the curve in a similar way that pH does, further amplifying the effect. Another great feature is the fetal hemoglobin, the ones we talked about earlier. Fetal hemoglobin have a left-shifted oxygen hemoglobin dissociation curve opposed to their adult counterparts. Thanks to this difference, oxygen can be transported from the mother's blood to the baby's blood via roughly the same process as in the lungs. If the fetal hemoglobin did not develop, babies would have a much harder time getting oxygen. Before we end this video, I will tell you about what makes carbon monoxide such a dangerous gas for us to breathe. Carbon monoxide binds with hemoglobin with 240 times higher affinity than oxygen, meaning that a spot reserved for oxygen gets taken by the carbon monoxide. And as if taking an oxygen spot isn't bad enough, the oxygen monoxide opens the rest of the hemoglobin much more than an ordinary oxygen molecule would. So the curve shifts dramatically to the left, causing hemoglobin to not release oxygen when it's needed.